talk to you about a lot of the things that we've been doing, not just the drones. Trying to give you a sense of just some different ideas, different thoughts, different ways of kind of addressing research in the classroom. Before I do that, I think one of the better ways to kind of introduce this all is to kind of explain who I am and why I do what I do and kind of how that fits in with this. So, my identity as a scientist was framed at a very young age. I would say about 3 years old when I decided I was going to be a whale and dolphin doctor. Family business was always in animals, my father being a veterinarian, my mother working in rehab, animal rehabilitation. So that's always been, animals has always been very important to us as a family. And where I grew up, upstate New York, right about there, you know, not a lot of dolphins there, right? So it's one of those exciting, mysterious things that you might be able to work with someday if you're really smart and can find a way to get where you're not, right? So yeah, it's a lot closer to the freshwater than it is to the saltwater. So that was always the mystique, right? You could maybe go to Cape Cod, we had Niagara Falls here, had the marine land, so you could go maybe see some of those animals, but you know, the idea was always to try to get to the coast somehow. And so I did that for my undergrad. I went to Southampton College, Long Island University, kind of an ironic little 40th anniversary celebration because the next year they closed. So it was one of those things where you had like 600 on-campus students, making it really difficult to maintain that as a marine biology program, which is kind of where I was focused. But we did have a really good marine station, and it was one of those opportunities where I had faculty who, I mean, it was really small class sizes, and you just had one-on-one with faculty so much that you got a really strong education, and so you felt really confident coming out of that. To give you an idea of how small it was, this is actually where I got my first teaching job, and not necessarily as a tutor. When I graduated, they lost a faculty member really abruptly, and they say, okay, would you like to be an adjunct professor for us? So like, I graduate and they say, hey, we have a position where you can teach students to teach animals to do that. Okay, so this is the experimental animal learning course, basically teaching students operant conditioning in animals. And what was unique about this university is that I could be trained as a biopsychologist. So my training is actually half psychology, half biology, which is kind of a neat interface when it comes to education. Because you can sit there and say, okay, why are you making these mistakes? Why are you thinking of it this way? You know, exploring the metacognitive sense of learning. So there's a lot that this little guy can tell you about reward and punishment within a classroom, because you really get a sense of where are the students enjoying what we're doing, where are they feeling reinforced, and where are they not getting that reinforcement, or, God forbid, feeling punished, right? And these were all the things that you wanted to avoid in animals, and these are the things, of course, you want to avoid in the human animals as well. So a lot learned there. And I took some of that to the University of Chicago, where I did my master's and my Ph.D. And you might be saying, that's way far away from the ocean. And you are absolutely correct. Because I came to work on some ground squirrels with Dr. Jill Matteo, in the Department of Comparative Human Development. Now, University of Chicago is an interesting place because they don't really have a biology department or a psychology department the same way. They have these really high-minded ideas about how you can integrate all these different thoughts. And this worked really well for me because I hate boxes. I hate being put in boxes, and I hate thinking through boxes. I like to move around with different ideas, right? You know, where's the line to this? I don't care where the line is. I don't want to think about the line. I want to think about where are the opportunities, where's the potential, right? University of Chicago is kind of a good place for that. So my initial master's work, we got to go out to this area. This is the Sierra Nevadas, so not too bad. If you're going to be away from the ocean, that's a good place to be away from the ocean. And what I would do is we would kind of trap up some of these little ground squirrels, and I decided, well, you know what you should do with a ground squirrel? You should put it in a radial alarm maze. And so there is a ground squirrel being put in a hand-built radial alarm maze where the animal has to look at the different cues in its environment and figure out which is its escape arm. After it escapes, it gets 5 minutes of freedom to just hang out in this little enclosure before it traps itself into one of our little traps. So this was what I did for my master's, basically looking at how these squirrels use different cues to navigate in their environment, and found that some of this stuff's actually heritable. These squirrels actually are born into different environments, and those are the types of cues that they learn. So if they live in a forest, they use trees. And even if their offspring are raised outside of trees, they still use trees. So it was a heritable, or at least there's a heritable component to what types of cues they use to navigate. And so that was kind of that later informed some of the teaching that I would explore in other places. All right, so at some point I said, you know what? I love the squirrels, they're great, but I gotta go back to my love. I gotta go back to what I really love, which is the marine mammals. And I had an advisor who was patient enough with me to say, okay, I'm not a marine mammal person, but if you want to work with dolphins, we have the Brookfield Zoo in Chicago, we have Shedd Aquarium, there's some ideas here. And so what I did is I embarked on what was fundamentally building a lab as a graduate student. Because the University of Chicago has no marine mammal infrastructure. I had to build that. I had to design that. I had to go and, when we did this project, it was first to go convince the Brookfield Zoo that I knew enough about what I was talking about that they were going to let me work with their dolphins, which these are not cheap animals as far as, public and displaying these, and there's going to be sensitivity here, right? So I had to figure all that out. One of the other facilities that I had worked with for this study was Disney. So when you're a young kid trying to navigate through Disney's legal agreements, that's intimidating, right? And so I had that experience dealing with this study because I had gone from different facilities to record dolphins' signature whistles. So this is a signature whistle. And this is a dolphin named Allison, Nolani, and that's the mom to Pico. And so if you listen, you'll hear their individually specific names. This is what they named themselves in their first year of life. So that's Allison. That's Nolani. And that's Topeka. And that's a whistle they will keep as their identifier for their entire lives. Okay?
Question.
Yep.
Do they come up with the specific frequency, or they have?
They come up with this. So they'll listen to different dolphins in the pod, and they'll say, I like that part, I like this part, we're going to remix, and we're going to mix and match. This dolphin here was a very people-centric dolphin, and that is the trainer's whistle. That's a 10 kilohertz trainer whistle. She just liked the parts of the human whistles. She took that and put it in her signature. So, yeah. So this is the work that I did for about 5 years in my PhD. And again, this kind of sets the framework for some of the cures I'm going to talk to you about. Okay, so this time that I'm getting my PhD is also around 2008. And so I don't know where you all were in 2008, but money was a little tight. So at the time I was living with my family in Louisville. I was ABD, so I didn't really have to physically be in Chicago. And I needed to go get a job. So I got a job as a substitute teacher at the Jefferson County Public Schools. And let me tell you something. If you want to do like boot camp for teaching, one of the things you can experience is trying to teach Spanish when you taught yourself Spanish the night before because you don't know Spanish, but it's a school that they're more concerned with whether that students kill each other than whether or not they learn anything. So I'm sitting here teaching myself Spanish, and I got a security guard sitting at my desk with his taser out, because that's the kind of classroom management we're dealing with here. And so I learned really quickly that the worst thing that can happen when you're teaching isn't as bad in college as what it can be in other places. And so that was really helpful because I never feared things like getting in front of a 300-person lecture hall after that. It's like a set point, right? It changes your perspective. So that was enlightening, I'll call it that. From there, I went to Oklahoma State University as a visiting assistant professor, and later on I would become a teaching assistant professor after a postdoc. And I met this wonderful man who's with us today, Donald French, who really taught me a lot about what I'm going to talk to you about today. He's the one who helped me take my incoherent and incogent ideas and make them into something that is understandable to the rest of the academic community. And so for that, I'm eternally grateful. My time at OSU came to an end, and now I've worked 2 hours away from where we are now. So that's how I got to where I am. And so one of the things I want to address here first is the role of the professor, right? And for everyone it's going to be a little different, but very few jobs can have all of these things as part of one gig. And in all the places I've been, in a lot of situations, I had to do all these things at once. And if you ask professors how they spend their time, well, there's a 1999 study, although I'm sure this is well out of date. What the departments really expect out of time here, about 175% research, 20% teaching, and service 20 there. And then how we'd like to spend our time is just don't tell us what to do, right? But at the end, this is a lot of what happens. We're tired, it's resource intensive out of us, and there's had to be some decisions made about what to do. We gotta allocate resources, right? And so what used to be very much a framework of combining teaching and research has become a little more like this in some places. And I've had this experience. I've had a teaching position where the decision was, well, okay, you can do some of this research stuff, but we're not necessarily going to support you doing that the same way. A lot of think pieces started coming out. This is in 2012, Ian Clark, where you want to get better research and teaching from universities? Here's my model. And he comes up with this wonderful model which says, okay, here's what we got right now with teaching and research output being at this kind of theoretical where really you've only got about 3 faculty in your department that are doing most of your research value. Just let them do all the research and everyone else can just do the teaching, right? And if you do that, look at how you optimize this. You go up from 1 and 1 here to this mystical 1.2. There's a reason there's no labels on these axes. This is all very theoretical. What is this quantity? The magical numbers, right? And these modeling approaches were really taken seriously as this is what you want to do. You want to have some teaching faculty, you want to have some research faculty, and you want to start to separate these things out. And that is a way to say, okay, now we don't have to burden you with teaching and research. But the question is now, when we look at this, what is ultimately happening? So the proportion of non-tenured teaching intensive to tenured research intensive appointments has risen sharply. The dichotomy between the two is becoming more and more clear. 70% of university faculty now are teaching focused in positions described as either casual, limited term, fixed term, part term, lecturer, visiting faculty, replacement faculty, which I think is a really horrible name, or adjunct professor, without a research component. We're splitting these things apart. Okay, so what's the cost? What's the cost of that? What does the increasing bifurcation of these situations do for both students and faculty? Well, Mihai Csikszentmihalyi, who is the author of the book Flow, who does a lot of work about student engagement, wrote a lot about this. And at the University of Chicago, he was one of the first to really take on undergraduates into his lab to do work, because he said, look, I want minds in the room who are not steeped in tradition. I don't want people who've only thought about this the same way for 20 years. I want new ideas. And you can get that from your undergraduates. And so he always valued the undergraduates as an intricate part of his lab and his productivity. Right? So that was one thought. Well, are we losing something from our R1 researchers when they don't have access as easily to the undergraduate source? From the perspective of the teaching faculty, look, they're happy to have you do some independent research with the students in many cases. But are they going to give you the time, the money, facilities, the space to do that? That's often a very open question. Now, we all know the value of cures in this room. I think that's why we're here, right? We know it produces effective gains, increased enjoyment for both science majors and non-majors. We know it improves career outcomes. We clarify to students whether or not they're really interested in this. It helps frame that, right? The other thing is, to generate the best results, novel to themselves, novel to the instructors, you really want to have these students become stakeholders in the field at an early age. You want them to engage in this iterative work, and they must troubleshoot, problem solve, because it's going to teach that resilience. And that's really what you want. You want them to go through the struggles, to engage the material in personal and meaningful ways, and to communicate their science in a professional setting so they really do get that full experience. Sometimes I joke with Don, we say you're cosplaying science a little bit. But in truth, the more authentic it is, the more the student really feels that they are a part of that process. And I genuinely love watching a student rise to the occasion of a presentation, of getting that paper. You get a draft back from them, and oh my goodness, you guys nailed that. They can do so much if you put them in the right framework. All right, so this is what you show. Of course, the bean counters. Yeah, this is it. We all know this, right? Here's all the literature's pretty good on that one. This is going to improve your student retention. But the problem is I need you also to support that from the administrative point of view. You can't just say, okay, it can't be just the stick. You got to give some carrot, right? So one of the things to think about is, okay, how do we support faculty supporting students in research? I think that's the next framework for this. And part of that too is also thinking about, okay, how do we support teaching faculty or teaching research faculty or research faculty to bring those two Venn diagrams closer together, back together a little bit without overburdening the faculty member? So how do we achieve this? Well, part of it is a course design, right? How do you want to integrate all of this into your courses? And so one of the ways of thinking about it is to help students with research. You want to help them generate some knowledge. You want to help them generate legitimate stuff that no one has yet seen. I thought when I was a young kid and people asked me, why do you want to be a researcher? I said, well, I want to be the first person to know a fact. I want to be the first person to know a truth in the world, and then I want to be the person who gets to find out who the second person is going to be. And that's a cool thing, right? You can give that to your students. That's an amazing thing to be able to do with them, right? If you introduce them into that, within their disciplines, this is also important because it helps them feel like the place that they're at knows what's going on. Some of us may come from kind of teaching intensive institutions. I think that there's some anxiety sometimes with these students who don't necessarily know that the place that they're at, not the big R1, is still connected with the world that they want to get into. And so you want to convince them, no. We're plugged in. We know what you need. We're going to help you get there. Okay. Obviously, you want your students to learn the research methods, and you want them to be motivated to learn with the direct involvement within the research. So that's part of it too. Okay. Now, students have to carry out some of that work, obviously. Students participate in research conducted by the faculty. I think that's really important. But it also needs to be, at some level, something that they generate themselves. It can't just be doing my work. They need to kind of come up with their own ideas, because that's really the genesis of a great scientist, is the person who can keep asking questions. Maintain job security, right? So, students learn and are assessed by methods that resemble their research procedures in the discipline. And I guess this is one that I really learned from Don, because he has the journals, and he would have students submit to the journal and go through a peer review process. My students will go through a peer review process. Their peers will judge their work, their peers will assess their work, they assess their writing. That's a huge part of this, right? Because you want students to get a real sense of the quality of not just what good science looks like, but how to evaluate that, how to think about that. Okay, and then you really have to learn about how student research is organized and funded. So that is a problem. So you have students who want to go and do independent research within the class context. Okay, someone's got to pay for this, and the university is not paying me to give you that money. So we do have to think about, okay, do we possibly go looking for grants? Is your idea really good? And we can think maybe we can do that. And I've had students work with me on some grant writing work. We've had other students where I sit there and say, okay, how can we fund this? How can we think about developing kind of a model for getting funding somewhere? So those are some ideas. So these are some of the things I'm going to talk with you about today. We're talking about some independent research we did in fish biology, some work we did in invertebrate zoology, animal communication, bioacoustics, so it kind of connects back to my old work, and then finally some of our drone work. So let's talk about fish biology. This was a class with 2024 upper division students, divided evenly between, let's say, natural resource focused students and students focused on biology. These are two very different audiences. One of them is about bean counting and kind of identification and recognizing what you've got in an area, management. The other is, how does the swim bladder work? Where does this, in the evolution between shark to Actinopterygii, what are the transitions and how do those operate? Very different audiences. And Cures is actually a nice way to bridge the gap here. This was a class with 2 1.5-hour lectures weekly with a 4-hour lab, and field components were baked in. The course as I received it, because in this case I was a teaching assistant professor. Here's the course you're teaching. Okay, got it. Taxonomy heavy. Some research based off the work of the previous faculty member, focused mostly on morphometrics, and a moderately high DFW rate when I got it. Here's what we did. Maintain, focus on some of the taxonomy, because we've got these natural resource students in there. They're going to need to know some of these IDs. But we did focus a little less on the identification of specimens. We introduced some empirical labs. Students would have to collect data, analyze those data, specifically some on mate choice in guppies. We introduced independent research as a part of the course. So now at the end of the course, the students are to submit a finished research project. Now these things have backbones, which means it's IACUC. Oh yes, so IACUC became a component of their project. You've got to get through IACUC. So we can't just electrocute no, you cannot just electrocute the fish. You have to have a, this is how this process operates, So yeah, animal care and use became a part of what they had to do. We had to get through IACUC. And we did maintain a lot of the field components for this. All right, at this point, these changes led to about a 5% DFW rate at the end.
What is DFW?
Oh, D's, F's, and withdrawal. So more students were successful with this course. Okay. Oop. Clicker went on its own there. So I'm going to talk you through some of the work that they had submitted, if this computer will work. Okay. One clicker. There it is. Okay. These were students, this was a specific group of students who was not a big fan of Dolphin Guy coming in and teaching them about their fish in Oklahoma. They were natural resource management students who were very skeptical. I'm going to be very honest with you about how they approached this with me. It was when we engaged this cure that things completely flipped around for these students. We had one student who was absolutely convinced that if he went seining with the current instead of against it like his like the research mentor had told him to do in his independent study, that he would catch more fish in the seine nets. Right? He was just sure of it. But this graduate student who was his mentor said, no, you are not. You are only to do it against the current like this. And he trudges very slowly up against the current. He knows in his heart he cannot catch as many fish because he's not moving as quickly, and the fish will just go swimming around his net. And he's just absolutely convinced. And I said, all right, let's do it as a study. And so they did this. They went out, they did all the different methods of catching. They went up against the current. Then they went with the current, chasing the fish down the stream. He was right. He was right. He caught more fish, more variety of fish, more diversity, if he went with the current and caught the fish by the back end. And that actually kind of fits, because if you start thinking about the visual field of the fish, they can see what's coming at them a lot better than what's coming behind them. The student was right. He was able to take this poster, go show it to the graduate student. So there's vindictive cure. This group of students did a really cool study where they used videos of predators, of guppies, and put videos essentially on little iPads on the side. And the fish vision was pretty good with the iPad. This is kind of when iPads started becoming a little more ubiquitous. This is 2014. They found that the fish were capable of recognizing the evaluative threat of different predators. So both were predators on each side of the screens, but one is a faster predator than the other. So they would go toward the slower predator and avoid the known faster predator. These are fish that were born in a pet shop. And they knew how to swim to the right side to avoid the more threatening predator to them.
Is the actual predator image?
Image, just the image.
The video instead.
Video, yep. Showing a video. Yep, and controlled for movement, controlled for all these other things, yeah. Students had figured that out in this class. This group did mate choice. These are the kind of these Timbergian mate choice studies. Bill Rowland, who Don and I are both quite familiar with, kind of working in his lab at different points, would do these experiments with model fish. And you put the model fish in the pools and you look at which fish is preferred over another. And what you saw from these students is that the fish actually would use video screens to make determinations about which were the more attractive fish with the more gravid bodies or the more color or so on and so forth. And they basically showed you can get a lot of those same responses that you get in model studies with, again, video playbacks. And so that's what they did. And then finally, now I want to contextualize this. This was a group in 2014. 3D printing in 2014 is not what it is today. We had a student who, fantastic student. She had struggled a little bit with the course under the previous instructor. And she comes into it with us and just came alive. She develops the 3D image that they use to 3D print models. Those 3D models then get put into the pool and you let the fish choose. But now you've got this maximal control over all these different variables of body size and coloration and how, transparency. And these are controls that you never would have had in the old designs of just hand carving these models or using a little mold. And so she really opened up the idea of the potential to use these in a research context for these mate choice experiments. And so with the help of Don and his group, we ended up publishing this as a lab for ABLE, and it was implemented in the introductory biology course. It was scaled up. And so this is one of those opportunities for mentorship. So I have to give credit to Don here too, because it was really Don who says, hey, you're fish bio students who are doing a lot of this mate choice stuff. What if they were to come in and act as mentors to work with the intro bio classes? Because of course the TAs don't know how to do a lot of this, and and this is a lot of students. This is very big intro bio. So we had a huge acceptance rate. Almost all the students came back. They were done with the class, and they're coming back the next semester to help out and impart their knowledge to the next generation of students who would work on this as freshmen.
Did they get credit?
No, they did not. It was the pure enjoyment of being in that mentor position. It was self-reinforcing to some degree. So that was the mentorship component to that. So again, we scaled it up for the 1114 course, and then they were familiar with the 3D printing. The student's model, which she developed here, was scaled to be able to be used for the students. So the students could now decide in the intro bio class, maybe I want to change the fin a little bit, maybe I want to change the body size a little bit. What about with this color? What about that color? So now the students had control over those variables in their inquiry-based lab. Okay, let's talk about invertebrate zoology. So again, you're in a teaching assistant professor role. They come up to you and they say, you're going to teach invertebrate zoology. I say, I'm a dolphin scientist. I don't do invertebrates. Great, figure it out. So the course as I inherited it was about 40 upper division students, biology or ecology focused majors, 3 one-hour lectures, and a 4-hour lab. Taxonomy heavy. There's a lot of invertebrates to learn all that taxonomy. research proposals were a part of some faculty designs where students would submit a research proposal, but they didn't conduct that research. It was just basically a grant application. So for virtually no independent research, and they had a popular kelp holdfast lab where the students would kind of dig into this kelp bed that would be sent from Monterey Bay. So I kept that part because the students kind of like that. But there were no hypotheses or really tests related to it. Okay, so what I did, we maintained some of that taxonomy focus, but for the most part, we scaled that out. And this is the course where I said, students, as groups, you are to go out and get an invertebrate. Whatever invertebrate, you want to dig into your yard, pull up worms? There you go. You want to go order millipedes from somebody? Go ahead. You want to get into moths? Some of the students had access to scorpions. Just be careful with it, you know what your safety thing is with that. But yeah, basically we had students as a lab group would collect their own invertebrates for the course. And this is the invertebrates they would keep for the whole semester. Okay. We introduced empirical labs. So we did some version of it where, I should say fish behavior, this is invertebrate behavior. We did some kind of sexual selection stuff. But generally speaking, oh, there was no echo. This is what we did. So running through this, first thing they had to do is an identification. So basically like a taxonomy identification. Measure your animal, how long, do averages if they had multiple individuals. You got multiple hermit crabs, measure all those legs. We did some behavior work, ethograms, they would develop time budgets, they do comparative analysis between their animal and another team's animal about activity levels and things like that. So again, they're thinking empirically. Then we go into electrophysiology, and I'd say, okay, today I'm going to give you roaches. You're going to take the leg off the roach, we're going to stick pins in there, we're going to get electrical signals from those roach legs, and those are called spikes. And so basically they're learning about electrophysiology through the course of that. The next day, or the next week, now you got to do that same thing, but you've got to do it in your animal. Now, if you've got a worm, there's so much electrical interference, you're going to have to build a Faraday cage to get that same result. If you've got a hermit crab, that's a heck of an exoskeleton. You might need to bring a Dremel. We had one group learn about millipedes and their hydrostatic skeleton. The second you stick a spike in that, they bleed out. So the students had to figure out, we need to put putty down and then insert the spike with the putty in there. And then when you take it out, you basically have the Band-Aid there. It keeps them from bleeding out. So students had to learn that. And then they have to use neural maps to figure out where the neurons are that they can spike. So all of that is independently derived, and you have to come to class doing your homework ahead of time about what your specific study species is going to need to get those same data. And then the next week they did a comparative analysis between their electrical receptions data and another team's. And then we did a field project, spring break happens, we did a little bit of turbo tricks. And then, so as these labs get more and more intensive, by April 5th, welcome to our lab, because now you're going to act like grad students. Now you're going to be implementing your own research. So they had to submit a proposal at this point. They had to submit a research bibliography at this point, showing that they've done the reading. Okay? And at this point, there's a manuscript due. There's poster presentations that they do in class. All of this is generated with the students basically doing independent data collection for 3 to 4 weeks. We are taking over the entire building at this point. We've got students with, scorpions in one room working. We've got tarantula teams in the other looking at how tarantulas move on different terrain surfaces. And over the course of my teaching of this, we had 24 different independent projects done that the students generated themselves, which meant a lot of homework for me. I had to learn a lot of this stuff because the students are coming up with these ideas. Like, okay, what about tarantula feet? Okay, I have to think about that. And so I wanted to kind of highlight some of the rubrics we use. I'll make these slides available so you can kind of see how we would do this, but we looked at your rationale for, we looked at hypotheses. We really want to focus on what are your hypotheses, how are you testing them, what are your predictions, what is your research approach, is it feasible? So there's a lot of checks in there to make sure that the students are really thinking through these things. So when they sit down to do this, again, 3 weeks isn't forever. So you have to make sure that they're ready to go when it's time to go. And then I actually wanted them to think about the broader impacts of what they were doing. We had rubrics for their bibliography. So again, I'm not going to go into super detail here, but how, what does your bibliography look like? Did you format these things correctly? Did you find research that specifically speaks to the hypotheses you're talking about? Okay. And so, then we did a poster rubric, and I did absolutely crib this from Don and what he does with HHMI and the Life Science Freshman Research Scholars, because that was such a good poster. Poster rubric, because it really does kind of highlight what is to value in a poster. So I'll leave that in there so you can see that. And then finally, this is the manuscript rubric, where we kind of lay out, okay, what did you have in your intro, your abstract, your methods, results, the presentation of your data. Did you have your clear figures? Is it connected to the hypotheses? Did you do the hourglass approach where you got down to a thesis, and then from your discussion, did you start very focused on the results and move out to a broader context, and then push the references? All right, so let's talk about my Scorpion team. So Scorpion team, first I was always a little nervous. Okay, hey, you got Scorpions. He's like, we know how to do this, and they showed me they know how to do this. So I was like, okay. What we did is we borrowed from Don some of these light systems that are used for algae and growth, plant growth. And because you can control what color lights and wavelengths of light are present. And this became a sensory physiology or sensory behavior lab, because what they were doing was they were putting the scorpions in these different light chambers and looking at activity levels. And they found that scorpions are seeming to process well in the blue range. That's where the scorpions are confidently moving around, even more so than in white light. So if you put your scorpion in a completely blue environment, that's scorpion visual prime time. And so they move really well through the environment at that light frequency. And so
There's scorpions that show up under black light.
What's that?
There's some scorpions that show up under black light.
Mm-hmm.
Are they scorpions that show up?
I'm not sure if this species does. I'm not sure about that. But this is the this is their Asian forest scorpion. But yeah, blue light. This is my death heads moth team. This poor team. They started with banana slugs, and everything they were doing was not working. And they gave a really good presentation in class on the death heads moth. And I said, y'all have a really good project there, but we can't get death heads moth. They live in Europe. It's like, don't worry about it. Here's what you got. This is the sound this thing makes, and nobody knows what this sound is about. Nobody knows what the function of that was. And so we said, what about a hypothesis related to bat anti-predation? So we know that some moths have anti-predator responses to bats by basically releasing jamming signals. And so what this group did is they looked at how that communication of the death head moth maps onto bat calls. And the students at no point had to handle any animals for this. They looked at it purely on the spectrograms. Then they went and did playbacks, and we thank the engineering department because they let us use their soundproof rooms to do these kind of playbacks where you would manipulate where's the death head moth, where's the bat, where are the different areas where you could have these masking effects where the sound cancels each other out. And it turns out, and these are really kind of weird to explain, but here's the moth spectra, here's the bat spectra, they're pretty much lined on top of each other. And when you see them lined on top of each other like this, there's a strong opportunity for this masking effect. And so they're able to, so there is a strong evidence here that that's what that call is about. And so again, students figuring that out. All right, so let me talk about mammalogy here. This is a course that I had taught across 2 universities, OSU and SFA. The OSU one was in much better shape. The SFA mammalogy was not where it needed to be at all. The course in both places is a 43 student upper division kind of thing where you had biology and again wildlife management. Again, 2 very different types of students. 2 very different focuses. 2 1.5-hour lectures weekly, and then that 4-hour lab broken into 2 sections at SFA because unfortunately the room we have isn't even big enough to fit all students in it at the same time. So I have to have a virtual version of the lab on one day for 20 students and a in-person version of the lab for 20 students. And they have to flip A and B because we can't get a lab room that's big enough. So that's another thing you have to think, right? There's that kind of limitation. So the version of the course I had, at least at SFA, incredibly taxonomy heavy. In fact, all the students did was work with dead specimens. Half the time, there was not even direct instruction with the students about what they were doing. They just had dead specimens, and that was that. That wasn't helpful. The course was very unpopular. On average, there would be maybe 5 students enrolled in the course. No independent research, no organization, and it was all pretty much lecture. Changes we implemented. So, we maintained some of the focus on taxonomy for the management aspect, but we entered some inquiry-based labs. I'm going to talk about a few of them here because I wanted students to at least start the process of what is data collection. Some of these management students have not had any of that, had no concept of hypotheses, data collection, good study design, and they needed that. So I introduced some empirical labs on drones and bioacoustics, which we'll talk about, and I implemented some field components. Got it down to about a 5% DFW rate. So this is the concept for bioacoustics. And again, this kind of ties back to my original training. Students learning aspects of mammalian bioacoustics, including analysis of acoustics, but also playback studies. And I had to, my management students were like, why do I need to know this? And I had to teach them about how we operate with marine mammals. Marine mammals are a very difficult species to find sometimes. They're underwater, and some of them can go very deep for a very long time. And so we've gotten good at passive acoustic monitoring. So I said, if you want jobs in the 21st century in management, you better start getting good with acoustics, because now we can leave out the acoustic beacon, it records the animal, and a computer can tell you which animals walked by without even needing the camera trap. Can go for a lot longer than a camera can. All right? So, okay, well that's going to be relevant to my, yeah, it's going to be relevant. Okay, oh yeah, I'm good now. So, students work in groups of 4, and they're given a kit to record and do playbacks with. So here's the elements of that kit. And so I'm going to show you some expensive drones, but I wanted to show you something that didn't involve a mortgage. So, these are the kits. This is a Hospel shotgun microphone. Okay, this is $40 on Amazon. It has a little eyepiece in there so you can actually see the animal that you're aimed at. All right, they can record at any distance, and it's pretty good for what it is. Now the system doesn't. It has a little memory card. It can have a little bit of memory into it, but we got some of these. You can get them at Walmart. This is a kind of an expensive one, but you can get some of these at Walmart for $40. They're just little recorders for a classroom. Put a little memory card in there, do your microphone in from this to this. Students hit record here, they just have this on their pocket, hit record, and then when they press this little trigger button, it records, and they can hear it right out of the earphones plugged into this. So students are able to listen and record what they're getting, what they see in their little scope. Okay? Great little device. Then, $10 at Walmart, here's your playback speaker. Okay, the students connect this to their phone for Bluetooth. The fidelity is good enough. Hide that near a tree somewhere. You walk off. I'll show you exactly what to do with it. Okay, so in lab, so this is my in-lab students. They're trained to analyze animal vocalizations and even learn what we do for signature whistle sorting methods in dolphins. Why? Because they can appreciate the automated methods when they have to do it by hand. So they see the value of what we call PAMGARD, it's an automatic sorting method. Okay, and then students independently determine how to best collect the data in the field, because I made it a little contest. Who can record the most species diversity using those tools in acoustics? And you need to be able to show that these were all different species. Well, Dr. Bruck, how do I know that it was what species it was? That's where you start having to do those acoustic comparison systems with, again, these automated learning platforms that can explain, okay, This is what a lot of the apps do for bird people. It's the same type of thing. Yeah, exactly. It tells you what species it was based on these algorithm comparisons. Students get to do that part and say, okay, now you know how many species you've actually recorded. All right. Nobody was doing anything like this in any of the classes, and to this day I haven't found another mammalogy class that's doing anything with these types of acoustics. Okay, so this is just kind of a lineup of the handouts that they would get, but basically it teaches them straight through how do you do this. So for the students that are in the field who don't have me standing right there with them, it really highlights what you need to do. It shows what the acoustics are. It lines up, okay, this is what you would expect to see in this species. If you're not seeing this in your recordings, something's wrong. All right, then contact me. So they know what to look for. All right, so this is the ground squirrels I work with Dr. Matteo with at the University of Chicago. And what I said to the students is, let's do this as it's a research experiment, but it's going to be a little more controlled than what we're doing in inverted fish. I'm going to have you play alarm calls of a different species to the squirrels on campus. That's the ground squirrel alarm call for a coyote's on the way. That's the alarm call for big scary raptor's going to come down and eat me. That's the gray squirrel call. Okay? So that's what they are alarming about something. What I had the students do was test this idea about something called affect induction. The idea is that can these calls from a different species hijack the nervous system response of a different species so that they will respond with an anti-predator response, basically posting. Right, that's what they do. And so the students would look for this, and you know what they found? Actually, yeah, there's a lot of this posting behavior. We had control with very similar sounds, but control sounds. There's something about the Belding's ground squirrel alarm call that gets skurrids to do that. So it actually supports that affect induction, where it's not necessarily referential, but it hijacks the animal's response system. Okay, and so on the dolphin end, this was really cool because this actually led to students really being able to get out in the field. So I took a lot of signature whistles from my research, put it together as a lab. This is what a dolphin signature whistle is. This is how it operates. To this day, there's no other class that's doing this type of work. But for the students who, hey, you know, I wanted to work with dolphins with a kid, I didn't think I'd ever be able to. And I went to SFA, or I went to OSU, or I went to these landlocked places, and now you're here, and you can help me work with dolphins. They got to go out and actually record those very animals that they had worked on in lab. So these were students from my mammalogy class. We took them up to Chicago. They were able to do the recordings when we had another dolphin come back to that facility after 16 years. And the students who were trained in that lab to figure out which whistles were which were able to sit at these computers while I, as a proud parent, taking pictures. They knew exactly what was being whistled all around those pools because of the training they had in that class, and they were able to generate their own research based on signature whistle production in relationship to the introduction of a new dolphin to its former pod in that situation. And so they were all very excited about that. Long drive, though. Long drive. Okay, let's talk about the drones.
My name is Dr. Jason Bruck, and I am an assistant professor of biology at Stephen F. Austin State University. The study that we're working on while we're here in Bermuda is part of a comparative study looking at how dolphins in different populations respond to UAVs or drones. And the idea is to look at how the dolphins respond as the drone gets closer and closer, because as we use these devices in the wild, we really don't want to shape any of the behavior from the animals there. We don't want to have any real change in response. So part of this initial project is just looking at how low can the drone get before the animals start responding. So while for this project we're using off-the-shelf drones, eventually we're going to build our own drone that's going to allow us to collect the hormone samples from dolphins as it flies very close to their blowhole, and when they come up to breathe, we collect that sample. So part of what we're doing today also is collecting visual images of the dolphins when they breathe, and so we call these thermal images, and these thermal images are giving us a picture of what that breath field looks like, and it's going to help train our new drone how to collect those hormone samples from animals in the wild.
I'm Savannah Damiano, and I'm a graduate student at Stephen F. Austin State University. The ultimate goal of this project is to develop a new drone that can collect health data from wild populations of dolphins in a non-invasive way.
If we didn't have zoos, and we didn't have aquariums, and we didn't have facilities like Dolphin Quest, we'd have to invent them. Because without facilities like Dolphin Quest, who work with researchers to develop new technologies and new conservation methodologies, we would be lost.
I have to mention is a lot of the work that I do is really based on the partnerships I have with a lot of these facilities who let me bring students to these animals. And they let us do this work to teach students about those animals and to give them those experiences that you really couldn't have anywhere else. And so UAVs, or uncrewed aerial vehicles, are the new technology that is making marine mammal science really move much quicker than it ever has. It's non-invasive, we get a great view, reduced animal disturbance, and then with the infrared, we're now able to do really strong health assessments on these animals. We're able to look down right into their blowhole when they breathe and get a body temperature from 200 feet up. That's amazing, right? And so we've got this new sense of new technology, and we want students to really be in the front of it. So when I talk about combining research and teaching, these students are using my research drones in their classroom. They are, and that's why I say this is operating without a net, because the big one, that's 14,000. And they know it, the students know it when they're taking these things up, that this is to be taken very seriously. Because this is the real stuff. You're working with the real equipment out in the field. So this is our surveys. What we're looking at here are the dolphins looking at our drone, which they are, which tells us, oh, you can see this and hear this one at this height. We need to be up a little higher. Okay, so we know where our maximum get close to view is, so that when we go out in the wild and do this work, we don't disturb those animals out there. So students learn how to fly 3 different UAV types. Students are in groups of 4. They're given a map of campus, and they must develop a flight plan. They must also get their trust license, which I'll talk about in a second, and they have about 22 minutes, which is the length of a battery cycle. Drone can only be up for about 22 minutes. Team that gets their most mammals, most diversity, gets their name on the award and 5 points bonus on the next test. So we have an award that we do every year. The team and their names get put on it every year that they do this. So we have one team now. Okay. Their objective is to assess the strengths and weaknesses of the technology to develop their research plan. Each of these drones has advantages and disadvantages, which I'll get into. So students must get the recreational drone license, and then they must develop that flight plan using the campus map, and they have to submit that to me for approval. Students designate a pilot out of their team, and the rest of the team has to support that person, whether it be through spotting on campus, or doing some ground research about things, or in the analysis of the videos that they'll get. So that's what that is. Okay. And then the students get a copy of their video right then and there. So give me your computer, here's the memory, you get the video, I get the video. So we can both see what happened. Okay. These are the 3 drone types. All have advantages and disadvantages depending on what you want to do. This is the fastest drone. It has the highest resolution camera. It is wind resistant. It is also the sound of a howitzer. It is very loud. But it has dual operator control. So if you have one person who really wants to be a part of this but is super anxious about flying a drone, they get to operate the camera. Another person can be the pilot. So that's a nice way to have students participate in this who might have some anxiety about flying some really expensive equipment. This is our Enterprise Advanced system here. This is a thermal drone. Really easy to operate, has stealth blades on it, still a little noisy. But it also has speaker playback capability. So, if you like, and I'll show you how this works at the end here, this is a playback speaker if you want it to be. And you can go and do playback studies. You can go sit this, you can land it and have it be a playback speaker that you put in a remote area that you couldn't get to physically, or you can fly it and have it be a playback speaker that way. You just send your sound file into the controller and it sends the signal through the system. It's actually used for crowd control, but we don't use it for that purpose.
How much is it?
That one's 65.
Oh, 65.
6,500.
Oh.
A little more than that. But we do have the affordable. Now this again, this is the real stuff, but there are some other drone options that are much more affordable. This is 400 for the Mini. The Mini is very quiet, and what a lot of students would prefer to use. I was surprised though, lots of students used any of the drone types, so actually it was pretty equal. But for those who had anxiety about this, there were some, Mini was their choice. Because if that crashes, not so bad. Okay. Now, students would, this is their flight plan. Again, I'll make that available. And here are the students doing that work, right? So here they took off in a parking lot. They flew around to the kind of where the lake was. They thought they'd see some mammals over there. These students were in one of the parks that's on campus. Same thing here. These students decided to fly around the football field. I don't know what they expected to find there, but that was where they wanted to go. They did get some beautiful shots of the football field. I will say that. This is the training. So they had one lab day of training for how to, and every student flew every drone in the training. And so we had a lot of battery cycling and that, but it worked really well. This is across the soccer field. As you see drones just going up and down the soccer field. So, okay, so these are the reports the students would write. They'd have kind of a write-up, a verbal write-up of what they did. Here were some of the images of the mammals they found. That's the squirrel and the thermal. Somebody with their dog, and then they write down what their species were and how they identified it. So that's with the advanced. So this gives you a sense of kind of what that thermal drone can do for you. This is, by the way, this is at night. So that's your deer. If you zoom in really close, and this one eventually gets there, you can see the head pop up as it notices the drone. There it is. Hello. What's that? So, okay, we also did surveys with the students because part of this was a grant with the university to improve retention. So we did want to get a sense of some of these scores. Within the survey data, we found that I will put, I think my future employers will value my experience with drones on a 5-point scale, 4.8. I put drone experience on my CV, 4.5. I think I will value the skills obtained at SFA using drones, 4.3. Using drones as part of my SFA education helps me think more positively about SFA, 4.1. Courses where I'm allowed to use cutting-edge technology and real-life applications make me more likely to value my SFA education, 4.8, which I like. I'm more likely to stay at SFA if I know I'm learning skills that make me valuable to potential employers, 4.0. And then I see tech skills like drone operations as potential asset for my future career plans, 4.3. On the things where I was measuring the change from pre and post, I did enjoy using drones as part of my coursework at SFA. Went from 4 at the beginning to a 4.5. One of the things in the qualitative that I really found was I had way more fun doing this than I thought I would. This is way more fun than I thought it would be. And then just students who you never would have thought would have been in it. Dr. Bruck, can I fly the drone this afternoon? If that's okay, can I just fly? And oh yeah, we can fly the drone. I always say yes to that unless there's a rainstorm. Okay. And I feel that using drones in class will provide me with an authentic research experience, 3.9, 4.2. So the students in this case are really using the methodology as part of the cure there, where what is the right drone from the approach I want to take to this problem? All right. So let's close this up. Students active in research and thinking, teaching and research. Okay, sorry. Students active in research and research thinking are better prepared for what the next step is to be for them. Research drives perseverance. That's the big part of this. They struggle. You see them struggle, but when they persevere through that struggle, it's so important, and they feel so accomplished after it. And I really like that. And of course, it gives them that ownership. It leads to good evaluations. I'm not going to lie. There's a selfish part to this. rely on that one. And it leaves students with something tangible. They write this stuff up. They've used this stuff as writing samples when asked by potential graduate advisors. I know that. They said, thank goodness I did that in invert because I was able to submit it as a writing sample. Because the guy I wanted to work with said, I need a writing sample. And they used it. And then when writing is part of the assignment, gives students a chance, again, to collect that sample. So, I leave you with Matthews. We're getting another one of those strange habla español sounds. Love Far Side with that. And of course, I have to thank a lot of people who helped out with this, including Don, who's here today. Family, thank you all for everything you've done. And I thank you. If you have any questions, I'm happy to answer. If you want to come play with the drones, we have them here. You can't fly them in the room, but you can see the cameras.
Are you using lecture time for this as well? Or just
In my marine bio class, I do. I will take a lecture and say we're going to do something inquiry-based today because I don't have a lab built in for that one. But the rest of them.
But you have enough time for them to
Yeah.
do that just in your regular lab?
Yeah, we'll do different things. Yeah, but for the all this other stuff is all done in lab.
Okay. Wow.
Yeah. Really, this was revolutionizing all their labs. They spent so much time on taxonomy. It was now identify this, now identify that. Now, I just feel at this point that there's a lot of technology in identification. I know we do it with acoustics. You know, it'll tell you if that thing makes a sound. There's a computer program that can tell you what species it was. So, and then at this point, we're taking pictures and it's telling you what the scientific name is. We're getting to that point. I don't feel that training straight taxonomy all the time is preparing these students for the 21st century.
And do you have meetings with the groups to discuss their projects?
Absolutely, we do. Yep.
During the lab time or outside of the class?
It depends on the class, but for the most part, we were able to do it during lab time. So while they're running their electrophysiology, I'm talking to them about their proposals. So, yeah.
All right.
Anything else?
Did you present at NABT?
I did not, but I will. I will now. 2018, yes.
2019 in Chicago.
2019. Yep, Chicago. Yep, with the cockroaches. Yep, leg dancing cockroaches.
I'm like, yes, it is.
Yeah, leg dancing cockroaches.
I remember you saying something about the spray stuff.
Sure.
All right, so I'm actually I'm Jason's wife, but I'm a grant writer for Oklahoma State University, and I work very, very closely with our Center for Sovereign Nations, which represents the students who are Native citizens, and they repeatedly tell me that the cures are great for their students because a lot of times, and you guys probably already know this, but a lot of them are first generation students. They have a lot of not a lot of confidence in their abilities and then approaching faculty, so this is a way to get them into research and really helps with that science identity and saying, okay, yeah, I am a scientist, I am this, and they've seen great improvements with their students who have gone through courses and new curves.
I'm not saying
Like I said, this is kind of a selfish question, but I teach in a very unique situation in high school. We've had 2 drones donated to us. One that was crashed into a lake, one that was crashed into a tree. In the class that I teach, we fixed the one that crashed into a tree, like replaced the motherboard. My question was, what happens to these if you ever burn out a motor or break something off?
You're in trouble. Now I got a guy. I will say that. I got a guy who knows how to do this stuff. He's in Georgia, but he knows how to do it. Oh,
Are you sending them to me?
was that?
Are you sending them to me? I'm just kidding.
No. I'll give name at the end. Rob is the first name. I don't have his last name handy.
It's not so much about finding a place to fix them because we've
Getting
successfully
ones
got them
donated.
running.
Yeah.
I'm just wondering, so my plan was we have this drone now that we can fix and it flies.
Yeah.
And I'm trying to have students now build sensors like you have, but using like Raspberry Pi or Arduino technology.
Yep.
And if somebody crashes one of those, I mean,
It's a big
You'll
problem.
take it.
Try to fix it and see, and, you know, have something that already works like that would be absolutely amazing. I was just wondering, like, are there many people that use those types of drones, and what happens to them when they crash?
This- okay.
A lot of times back.
Oh, like other people as well, which you could source money.
Yeah.
Yeah, so you can, right, and so if these drones go down, there, we do have someone who can help repair. The main function of the Inspire 2 here, this is for movie making, to be very
Right.
honest with you. If you're gonna go fly around dolphins, they want you to have this one so that you can take footage and give it to Netflix.
Right.
So that's why that one's good. And the camera control is completely independent, so you can, be swiveling around with the camera where the drone is, wherever it needs to be. Is that used in classroom applications? Not to my knowledge.
Right.
This is probably the only other. This is a police drone.
Yeah, that one's
So-
pretty interesting.
Right.
Sensors it has on
So
it.
right, here's your thermals on here, your RGBs on here, full high definition situation there. Also has lights, night vision, and then the speaker system. If this goes down, that's not so bad. This is essentially the base is what's called the Mavic 2. The Mavic 2 is a pretty common drone. There's a lot of bells and whistles on this version of it, but if there's stuff that breaks off or parts can be fixed and replaced. As far as kind of sensors built in, when students fly with these things, all the safety modes are on. The drone has a lot of precautionary ability. It's got sensors all in it that prevent it from just flying straight into a wall.
Anti-collision.
Anti-collision is all over it. So there's a lot of modes on here that prevent you from doing something completely stupid with it.
Right.
That being said, things can happen. This is operating without a net. So it's always a risk.
Give me some cool ideas for things that we might be able to build. Like we built little speaker systems and stuff with Arduinos and
Yeah.
things like that in the past. So
Yeah,
maybe we'll build.
as long as you can keep the weight down.
Maybe
Yeah.
it'll end up being like an $8 drone.
Sure. Yeah, and that is absolutely a part of this. The custom built drones and that's becoming a thing. My suggestion for that is get a hold of the racing guys, the guys who build the drones for racing, because they're the ones who have an intimate knowledge of how to build this out of the spare parts. They don't usually have huge budgets.
Right.
They build their own drones for speed, and max speed for these legally is 99 miles an hour. They build it to 99 miles an hour. They can do that, and they do it with stuff that is laying around. So I recommend you get a hold of the FPV drone pilots. They're the ones who are going to know how to do that with you. Yeah.
F what?
FPV, first person view. They're the ones who wear the goggles, and they see what the drone sees as it whizzes around, flies through hoops and things. So, yeah.